Our next speaker is Dr. Oliver Wüseke. He is a biochemist by training and is now a postdoc in the Noblik lab in Vienna. He uses 4 brain organoids to study the early development of the human brain, but he's also initiated a project that aims to establish a toolkit for present and future cerebral organoid researchers, which will allow them to tailor the technology to their specific biological question. His talk is entitled Brains in a Dish.
Okay, thanks for the kind introduction. What I would like to do now is switch gears a little bit from an organ that we use to digest our free lunch to an organ that tells us that we really like free lunch. As a frame for the talk, what I would like to give you during this talk is an overview of what we've done in the cerebral organoid field, different techniques, different results that we've seen, without going too much detail into an individual project, but to really just give you a feel for what it's like to working with them and what you could potentially do with them. So that, you know, by the end of the session you might say, hey, this is kind of cool, I want to try it out. Brains in a dish, or how we can use cerebral organoids to study human brain development and disease. And so let's start with something important. I hope the picture doesn't put you off. This is the human brain, one of the human brains. You're currently using it to analyze and interact with your environment. And it's a very complex organ that consists out of about 80 billion neurons that are interconnected via synapses and form trillions of connections. So it's a very complex organ, and with it comes, with the complexity comes a problem, and that is that there's a broad spectrum of possible diseases that you can get regarding your brain. A very easy one to grasp is the stroke, so cerebrovascular diseases, where an interaction of your brain with another organ is actually causing a problem. But then you also get neurological disorders where individual neurons of your system are perturbed, and typical examples for this would be Alzheimer's disease or epilepsy. Then we've just recently seen a rise of these infectious agents that specifically target your brain as well, such as the Zika virus during early onset of development. And last but not least, we have this very complex psychiatric diseases such as schizophrenia, where the genetic background and the epigenetics are completely unclear, and it's rather a collection of descriptions of phenotypes than really a classification for a disease. Having a brain is great, but it also has the potential to cause some trouble throughout your life. Luckily, in the past decades, we've been able to use mouse model systems and other model systems to actually understand the function of the brain and the related diseases to it. But with the recent, more recent findings, there's one really big question that I think is becoming more and more apparent, and that is how much of what we find in the mouse can we actually directly translate into the human? Just to give you a very simple example, I would like to point out the case of the NDE1 mutant. NDE is a protein that's found in mouse and humans, and you can see that on the right here, in the human, it causes an extremely severe form of microcephaly. So the size of the brain is much reduced. Moreover, the gyration, so the folding of the brain that you can appreciate here in the normal case, is lost, and the brain actually turns lissencephalic, so flat surfaced. And if we now compare that to the NDE mutant in mouse, what we see is that the size reduction in the brain is much less apparent, and the brain of the mouse in itself is already lissencephalic. So the phenotype that we associate with the gene is much less severe than what we see in the human. And mind you, this is a single mutation gene phenotype, right? So as I said before, the complex phenotypes that we see for psychiatric diseases are much more diverse and will be much more difficult to translate from the mouse to the human. So what I really want to get to is basically saying that we need a human brain model. And with the recent advantages in being able to reprogram cells from patients directly into stem cells and then turning these stem cells into neurons in culture, I think we've made a great step towards being able to model these diseases in a much more specific manner. However, when we differentiate these cells in 2D cultures, right, we mostly get a mixture population of randomly associated cells. And we know that the complexity of the brain not only comes from the complexity of the composition of the neurons that are in it, but actually also by their architecture and how they connect to each other. So I think it's very, very safe to say that three-dimensional model systems would have a huge advantage over two-dimensional model systems, even though they might be a little bit more tricky to actually establish. And going into 3D and modeling the brain, I would like to mention Wüseke Sasai, who, in my opinion, really pioneered this whole field. And what he did, in contrast to what everybody else was doing, is that he started from stem cells, but then he didn't plate them out on the dish and waited for them to differentiate it, but he actually put them in a solution together and let them aggregate into what he termed these embryonic bodies, right? So clumps of cells that start to differentiate. And I think this was really a key step. A second very important part was that he added an extracellular matrix protein to it, indicated somewhere here, if I can see that, yeah, that would actually provide the cells all in this an architectural cue, so to say, that then led them now to say, okay, I've got a polarity axis now. And I think this is one of the most striking pictures from an early publication from him, where he was able to generate small optic cup-like tissues out of mouse ES cells. And yeah, a great step towards 3D tissues. However, this was all mouse. And there was a postdoc previously in our lab, so the Knoblich lab now has her own group in Cambridge, Madeleine Lancaster, you probably have heard about her before, who actually took a very similar approach to Sasai's and said, okay, we're going to aggregate these cells into embryonic bodies, then embed them in matrigel, and cultivate them in bioreactors for a longer time to allow for good nutrient supply. And this is basically what an embryonic body typically looks like. You've probably seen that before. This is after embedding. So you see really the embryonic body really expands, grows in size, and then actually starts forming these cortex tissues that Madeleine also named the technology after, which is then a cerebral cortex organoid. So again, you might see, if you go into the literature now, you'll see that many labs have developed many different approaches to generating organoids, but it always comes down to aggregating the cells and then putting them in some sort of extracellular matrix. So don't be put off by the different approaches that people take. I think there's many different protocols that you can use to tailor the organoid generation specifically to your needs. So, as I said, talking about cerebral organoids, we're talking about the cortex of the human brain, right? So the front part of your brain. And since I'm not aware of how many brain researchers we actually have in this audience, I thought about giving just a brief introduction of what the tissue architecture during development actually looks like. To keep things simple, I would like to focus on the three regions here: the ventricular zone, the intermediate zone, and the cortical plate. And in these zones, there are different cell types being produced and located. So for instance, in the ventricular zone, you'll see that there's a lot of progenitor cells that actually produce more progenitor cells, right? So these are the stem cells of the brain. But they also start generating neurons, which then migrate via the intermediate zone into the cortical plate. And the cortical plate is where the neurons basically reside, and later on during development form all these layers that the human brain is so known for, the 6 independent layers where all the action is basically happening. So if I think about a 3D model for the human brain, it would actually have to recapitulate this structure. And in her original publication, Madeleine was able to show that marking the ventricular zone in red here and the neurons in green, that this structure can actually be resembled in the 3D organoids, right? So I think this is really cool, and the first advantage already over the 2D systems, that you get the 3-dimensional architecture that is reminiscent of what actually happens in the human brain. And then in a more recent publication, she was actually able to show that this cortical plate, that is a key developmental step in the human brain development, actually shows up nicely, as you can see in the staining here. It's this fat band of cells that accumulate in one layer, which then later on will turn to make the multiple layers of the cortex. Yeah, so here you have the staining showing a similar thing. This is extracellular matrix on the outside, and then this fat band of cells lining right beneath it. But then there was also work from Gu Liming, who's giving a talk in this afternoon, who was able to show that you not only get cortical plate, but you actually also get different neuronal layers. So these are the different neuronal layers that we have and certain markers that we associate with them. And you can see that in this cross-section, those markers line up in different depths of the tissue, really suggesting to that, yes, we can get a cortical plate, and yes, we also do get layering. Mind you, of course, this does not by any means look as nice as if you would cut open actual tissue, but I think it's a very nice indication of that we could actually get there. So we have a system, and what do we do with it? And here I'm going to give you the classic example from Madeline's paper, but I would like to remind you that similar stories have now been published on different diseases with a similar flow of argumentation. And I mean, the recent Krickstein paper on the Timothy syndrome, for instance, is a perfect example that you might hear more later on in this meeting. So just to give you the concept of how this basically works is that if you can get patient-derived cells and reprogram them into stem cells, you can actually grow organoids out of it and grow and compare it to to organoids that were grown from a healthy control. And Madeline did that in her original publication, and what she found that early on during EB formation, these EBs actually look very similar in size, right? But then you look at day 10, and you can already see that what is a microcephalic brain in the patient before also makes very small organoids in comparison to the control organoids in the dish. So that got her very hopeful, and she looked into the tissue to understand what's going on there. And what she saw was that many of the progenitor cells marked in red here, basically the control organoids by day 22 were full of these progenitor cells, right? And very few green neurons, right? There's almost no green neuron sitting around here, but all are red positive progenitor cells. But then when she looked into the patient, she saw that there's a huge accumulation of these neurons and actually a reduction in progenitor cells. And this had long been suggested to be one potential cause for microcephaly, and to cut this whole very long and nice story a little bit short, I would summarize it as this. So they had a microcephalic patient that had a mutation in the CDK5-RAP2 gene, and what they saw then in the organoid is that the progenitor cells had a defect in their spindle orientation. And what you have to know here is that depending on the spindle orientation during mitosis, the cells will actually produce more progenitor cells or actually start differentiating into neurons. And so in the wild type case, there's a lot of horizontal divisions, which produces progenitor cells only, whereas in the mutant case, the spindle orientation was more vertically and started producing premature neurons. And so I think that really nicely shows that you can go from molecule to tissue defect to patient in one single study. And as I said, I think there's many great other papers that show a similar storyline for different diseases now that really emphasize how valuable this model really can be. But of course, this is old stuff. It's 2013, so you might wonder what has actually happened since 4 years ago. And let me tell you, a lot. So I stole this picture from a great review from the Alotta lab, who basically summarized, just for brain organoids, the different techniques that are out there and available for using now. And what I want to get, I think there's 2 points that you want to take away here. One, I think we've been able to generate different kinds of brain regions, right? So the brain itself is as we said, very highly complex and has different regions that you might want to be interested in studying. And there's also certain diseases that specifically relate to certain brain regions. So having a toolkit available that allows you to get, a midbrain organoid, for instance, or a specific forebrain organoid, for instance, really boosts the potential number of projects that you can carry out. And secondly, one of the biggest differences in approaches that we've taken in generating organoids so far is the use of patterning molecules. And you can see on the right here that the original publication from Madeline was actually leaving patterning factors out, leaving the tissue completely to itself, allowing it to generate whatever it wants. And I think this has a certain strength to it because the cells actually will start signaling themselves, right? I mean, cells are like little computers, and when they lack a certain environment, they will produce this environment. And we've seen this now time and time again, but we actually see that different cell lines that you've generated behave differently because of their genetic backgrounds. So something to keep in mind is whether you want to add patterning factors or not have patterning factors. So if you want to be more specific for brain regions, people now tend to add patterning factors. And I think there's a lot of knowledge regarding this already from the 2D experiments that have been carried out before. Okay, so if I think of the brain, I think activity. I don't know how about you, but what really gets me excited is if I see neurons fire. And so when I started in the lab, I actually wanted to recapitulate some of the experiments Madeline had done early on and see if we can actually see neuronal activity. And one of the first experiments I did was to just label the organoids with this fluo-4AM dye, which basically reacts with a fluorescent burst when calcium shoots into the cells. And we know that early on during development, calcium signaling is very important for the neural networks to be set up. And so I'll just show you the movie because I think it's much more striking than me talking about it. And what you can see here is that there's a lot of neurons actually bursting with calcium activity over time. And that to us really suggested that, yes, calcium surges are definitely present in these organoids. Now, mind you, this is a live recording and it has not been sliced, right? So we're actually looking at the outside of the organoid, right? So this is probably why you do not see this layering that you would typically see what we show you in the stainings, right? So penetrating into the tissue with a microscope is right now still very problematic because the tissue is very, very opaque. So this is literally only the neurons that sit on side of the organoid. So I think that's kind of cool, but then actually the Alotta lab just recently published a paper where they drove the thing to the next level. And what they did was that they cultured organoids for up to 9 months in these bioreactors and then took them out, anchored them on a plate and shoved an electrode in it to probe for actual electrical activity. So what I showed you before was calcium activity, right? That might not relate to electrical activity, but now here we actually get electrical recordings out. And what you can see is that they indeed did detect so-called spike trains, so signature of action potentials. And they, moreover, in the analysis could show that these action potentials are highly correlated in between cells, suggesting that these neurons actually start talking to each other. And they didn't stop there. So what they also noticed is that some of the organoids contain pigmented cells. And when they looked at the RNA-seq data, they found that there's a lot of actually mature photoreceptor cells in these organoids. And so they thought, which I think is a fantastic experiment, right? They thought that if we can stimulate them, right, we should see a response of the organoid, right? So it's literally the first experiment where you feel like you're interacting with an organoid. And so what they did is that they took these organoids that had these photoreceptor cells, patched an individual cell where they recorded the spike rate, shown down here. And these are just 2 examples. And then they pulsed the organoid with light. And you can clearly see that there's a response of these cells to the exposure of light. Right? I think this is absolutely brilliant. And moreover, they actually saw that not only did individual cells respond, but if they shine the light on a specific part of the organoid, the whole organoid lights up with FOS expression. So this immediate early response gene that you would expect if you get neuronal activity. So they in the end suggested that most likely their organoids have highly connected neuronal networks that spread the information of being exposed to light throughout the whole organoid. So of course this is not as cool as what we can do in mouse at this point, but I think it's a clear demonstration of, this has only been 4 years, it's a very young field where this is really potentially going. And the last thing that I want to mention here, because I also found it a fascinating idea, is the BYOB approach, as I would term it, so build your own brain. And that originated from the problem that if you have an organoid and you want to study the interactions between different brain regions, you have to somehow make sure that you get the right regions next to each other, right? And in the original publication, you would actually have certain brain regions being located right next to each other, but the occurrence of these events was not ideal for quantification, so to say. And so the build your own brain approach is rather more about getting the tissue that you want, all the multiple tissues, and then locating them right next to each other. And there were just 2 publications, one from our lab and one from the ISSCR lab, that did exactly this. And the idea behind this was that during early development, it's very important that interneurons that are generated in the ventral part of your forebrain actually migrate into the dorsal part of the forebrain. So this is a process that is so important for the development of the brain that since your excitatory neurons are all generated in the dorsal forebrain and your inhibitory interneurons, right, are only generated here, that only functional networks can form if the interneurons migrate up. And there have been many implications that this migration is affected in diseases like epilepsy, for instance, or schizophrenia. And of course, it would be fantastic to be able to see this live and being able to study it in human brain organoids. So the idea is very simple. You generate dorsal forebrain organoids, marked in red in this case, and ventral forebrain organoids, marked in green. And then it's literally just putting them next to each other. That's it. You generate these balls, you put them next to each other, you cover them in matrigel, and then by day 60, they'll start fusing. So what our lab and the ISSCR lab then did was basically to look at migration from, in this case now, a green ventral organoid into an unstained dorsal organoid. And what you can see here is that there are clearly cells coming from the green organoid that migrated into the dorsal one. And I'm not going to show you all the quantifications here, but basically what they were able to show is that these are all interneurons of different subtypes that actually migrate into this tissue. And there's many fascinating observations, such as that they do not migrate into the progenitor zones, for instance, but they really stick to the cortical layers. So I think it's a fascinating, cool technique, and seeing it work so simply, I think really boosts my confidence in this system. Now, one very cool part about this is, of course, you can not only put brain regions next to each other that belong to next to each other, right? But you can also put brain regions next to each other that do not belong next to each other. And so there was a first experiment, actually control experiment, where Josh, in this case, from our lab, put 2 dorsal organoids next to each other, right? So these are the same kind of organoids, and we wouldn't expect any migration. And in fact, that's also what we see. So we did not see any migration from dorsal to dorsal, only from ventral to dorsal, as we would expect. This is a cool concept, right? Because the next thing that I want to point out is what you need to consider, if you want to venture down going into cerebral organoid research, and that is that organoids are in vitro systems, and not animal model replacements. There are certain advantages and disadvantages for both systems, and the downsides of the organoids right now, as I see them, is that vascularization still is under development. Some labs are working on vascularizing these organoids, but of course the mouse brain is getting vascularized, and this might have a huge impact on the development of the brain. So if we don't get that, we're limited to what we can model. Organoids currently also model early stage human brain development, so you want to pick, as the last point suggests, pick the right biological question for the system. And of course, behavioral studies are still somewhat difficult. So what I would like to end on is the positive things about organoids and what they give you in contrast to animal models that really play on their strengths. And first of all, of course, you can generate them out of patient-specific cells. You can mix and fuse the different regions, as I showed you, but you can also mix and match different cell types, right? So you could even think about classical in vitro experiments where you have spiking assays, so to say. So you get a cell that is extremely unhealthy and by itself would never form a tissue, but you could actually mix it with healthy cells and then study, because those cells would then take over making the organoids, you could study the defects that these cells actually experience. And last but not least, I think this is coming up in the future, these organoids formation of the organoids, maintenance, and analysis will definitely be subject to automation and tissue engineering. So yeah, I hope I could inspire you a little bit to go down that road, and I would like to thank STEMCELL Technologies for hosting this and giving me a chance to speak here. I would also like to thank all the people that contributed to the work. I mean, I literally just showed you one data slide from me and the rest was all from other people. There's actually a lot more stuff that I would have liked to mention, but I didn't have the time to. I hope you get a chance to check it out today at the meeting. I would like to thank the Knoblich Lab and my funding for giving me the chance to do this, and I'm happy to take questions.